Hey everyone, welcome back to the Wild Health Podcast. We're a genetics-based precision medicine company that's exploring all the big questions around holistic health, wellness, and performance optimization. If you're tired of the traditional healthcare system and want to sign up as a member with us, head over to wildhealth.com. And if you're someone who's looking for a high-touch, all-inclusive concierge medical experience specifically designed for executives and high performers, check out our premium program at wildhealth.com/premium. Now let's get into it.
Hey everybody, welcome back to the Wild Health Podcast. Mike Mallon here. So excited today to have Dr. Lasky-Su with us today to talk about epigenetics. Dr. Lasky-Su is an associate professor at Harvard University and a pioneering force in multiomics. Her innovative work in integrative metabolomics merges various omics, including a keen focus on epigenetics, to dissect complex diseases. She has significantly contributed to understanding the epigenetic factors influencing a wide array of conditions from cancers to neurodevelopmental disorders through over 225 peer-reviewed publications and more than 40 national and international presentations. Recently, Dr. Lasky-Su has delved into multiomics and aging research, utilizing the Mass General Brigham Biobank to explore environmental impacts on aging. Dr. Lasky-Su is currently leading several NIH R01 grants and private grants focused on epigenetics and metabolomics, further highlighting her role in advancing epigenetic research within the broader metabolomics field. Dr. Lasky-Su, thanks so much for coming on the podcast.
It's exciting to be here. Thanks for inviting me.
Absolutely. So I want to talk today about epigenetic age testing, and maybe even beyond that a little bit. I'd like to talk about epigenetics in general and kind of the future of epigenetics. But let's start with just sort of introducing the concept of epigenetic testing to the user. Can you provide us with a simple explanation of what DNA methylation is, how these aging clocks work, and maybe a little bit of its significance in relation to health?
Yeah, no, I think these are all great questions and really important ones as we're all thinking about our future and thinking about all the different ways we can generate molecular data now to inform our health. I guess I'll even start back up with the idea, right, of aging, because I think when we thought about aging in the past, I think there's been this frame or shift of how we think. And we, rather than thinking about just chronological age and birthdays, right, we're thinking about how are we aging and can we age better? And by that, I mean, can we prevent what we see as aging, right? And so in thinking of things this way, really the framework and how we think of aging is now, in my mind, I think the way I like to describe aging in the broadest framework possible is sort of the overall totality of someone's health, right? And if we think about aging in that framework, we're thinking about how every aspect of our body is aging and how, and when I say aging, I'm talking about sort of potential decline in ability, right? And when we think about that, right, that then automatically contrasts with this concept of chronological age and birthdays, right? And so the dynamic nature here is, well, from a chronological perspective, we're getting older. Can biologically we stay young, right? And that is a really important concept because we all want to live long and healthy lives. And the question then is, well, how do we do that? And are there ways we can actually measure how well we're doing that, right? It's one thing to actually do it, but can we assess how well we're doing that? And that's where epigenetics comes into play, and I think is so important because really epigenetics is sort of encoding how we are aging biologically. And this is a super important concept because I think if you went back, let's say 20 years or more, right, when we thought about aging, you'd always think about it from this concept of, it's genetics. And then, of course, there's your environment, what you do, are you healthy, do you smoke, et cetera. But, we always in the framework thought of this as genetics, that there's really this 100% hardcore thing of this is how we are encoded to age. And some people are way, have a big advantage and others don't. And while to some degree that's true, what we've discovered is that the epigenome is really this way where we look at our underlying genetics. And when we read that genetics, the epigenome is sort of reading what our genetic information says and telling our body what to do. The problem with what happens when we age is the way we read our genetics, and you can think of this as like an instruction book. Mm-hmm. slowly becomes a little jumbled. Words in the instruction book become missing. Sentences become fragments. And essentially that is how we can think of our epigenome, that the instruction book of our genetics when we age is slowly changing. It's getting older. Pages are ripping and this and that. And that's what's happening. But the question is, can we prevent it from happening? And can we even actually reverse it? And so what we can now do in the framework of epigenetics is look at that instruction book and sort of measure how well we are aging by how well that instruction book can actually be read. And it's a really amazing thing if you think about it. And what's even more cool is with epigenetics, not only can we get a big picture view of like, oh, what is our biological age? We can actually go all the way down to asking questions about what's aging well and what is not aging well. And that's a game changer, right? That's a major game changer, because if we now know what is aging well and what's not aging well, we know what we can target to, on a personal level, to age our best. And that's really cool, right? Because I always say this, we are as old as our worst aging part. So if I have a great heart and I can run marathons and this and that, but my cognition is really going down, I am as old as my cognition, because that's going to be the thing that's holding me back. And so when we look at this, if we can actually identify what are the things that we're worst at and how can we improve the aging of that system or even help reverse it to become younger, then we're really talking about not only long lives, but long, healthy lives. And that's what we want, right? We don't just want long lives. We want long, healthy lives. And so that potential is all there encoded in sort of our epigenome, which is super cool. So epigenetic clocks can tell us a lot of information, and starting with just our biological age, but going into all these other areas, which is super, super exciting.
That's awesome. I love the description of the metaphor of the instruction booklet. Like we're trying to put together a piece of furniture or something, and we start off, we're young, we're teenage years, we're like restoration hardware instruction booklet, and then or like Pottery Barn, and then like, middle age, you're like getting an IKEA instruction booklet, and then old age, you're like trying to follow the instructions from an Amazon package.
Exactly. Or those where like you get it and it's just like a sentence, and you look at like 5,000 pieces and it's a sentence, right?
Right. And a picture of the finished object.
Yeah. Play.
Walk us through the evolution of how these clocks have changed over time. I know we're on generation 3 now, so there's been a couple of rounds of evolution here.
Yeah. And what's actually pretty cool about this? I remember sitting in a conference, and this is a conference at the American Society of Human Genetics in Florida many years ago. And I was sitting there and Steve Horvath was presenting, and it was the first time I saw him present. And it was really the first time I think he was actually, in a major setting, presenting on this strong correlation that you can observe between your epigenome and age. And at that time, it was just your epigenetics and your chronologic age, right? We weren't even thinking about this concept of biological age. But I sat there in the room and I remember just thinking my jaw sort of dropped to the ground, because I'd been working in genetics for so long. And there are so many things in genetics that like we barely would, there are just these small effects. There wasn't anything that profound. And I looked there and I saw this amazingly strong correlation between your epigenome and chronological age. And I just sat there, wow, this is unbelievable. And so the whole concept here, and while people knew about it before, I think Steve Horvath really put it in this concrete framework of, look, we can estimate age from your epigenome. And it started with just chronologic age, right? And that was a great first step. But then what evolved was all these other ways that we could think of age, right? Going from, okay, can we just estimate it chronologically? Well, that's where we know biology comes into play, right? Because I can give you 2 65-year-olds who biologically, one may act like they're 40 and one may act like they're 85, right? And so that really got into this aspect of, well, can we actually do better than estimating chronologic age with the epigenetics? And so we got into a framework of looking at ways that we could estimate other metrics that could better describe biologic age, And in that wave of what came about, there are different ways of like, oh, well, we can estimate a metric of biological age this way or that way. And along the way, different ways of measuring it, came to light. And I think the one that, definitely even today still, I think, is considered sort of the gold standard of this generation of clocks is it was called Grim age. And that was, estimating sort of a metric of time until death. And the reason that became so popular is, because honestly, if you know the amount of time you have to be alive and you just invert it, that can give you your biologic age. And so that became, I think, and still today is been a very powerful way to look at biological age. And also, one of the things about these metrics are, okay, I can look at my biological age, but what does it mean? Does it actually have meaning? And when we look at these metrics of biological age, what you can see is they're more strongly correlated with disease outcomes, like things like cancer and diabetes and heart disease, than if you look at chronologic age, They have more biologic meaning with what actually happens when you age. And that gave us a lot of confidence that, okay, these are important. And evolving from there, in sort of a generation, I think now the third generation of clocks, came this concept of, okay, biological age is one thing, but can we actually look at metrics like how fast we're aging and age acceleration? I think this is a really critical point, because you can look at your biological age and that's going to tell you one thing. But my bigger premise, and I always say this, there's two things that are important when you think about biological aging. It's your current biological age and your rate of aging. And I'll give you an example why. So, third generation is focused on things like rate of aging. But I always like to think that the critical thing is second and third generation together. Because second generation says, okay, this is your biological age. A third generation is going to give you your rate of aging. But let's say, I want to give two examples of where these two things are important. Okay, let's say, I am 85. My biological age is 85, but I have a really, really strong decrease. My biologic age is reducing really fast. Well, that's really important. That means, okay, maybe my biological age is bad, but I'm getting better really fast. That gives me insight into I'm doing the right things, right? But it's a combination of those two metrics. Okay, now your biological age is super high, but look, you're doing the right things to get back on track. For let's say this person's 65.
Right.
Now let's say that there's another person who's 65 and their biological age is 45, but their rate of aging is maybe constant or maybe going down a little. Well, even if it's constant, that's pretty darn good, right? Because they're 45, even though their biological age is 45. If they can even maintain sort of that, they're still maintaining a drastic decrease in their overall biological age relative to their chronological age. So I always think the combo of the two things is really the most informative because it gives you an insight into where you are now and where you're going. And so I think that's really, a framework of how we're looking at it now and in terms of, looking at different individuals and how they can, where they are and where they're going.
Yeah, I can see how that's super important. I mean, you want to understand how you've been aging previously in your life to understand what your current set point is. And then you understand what you're currently doing is affecting your future rate of aging, right? Or I guess it's not necessarily future, it's current rate of aging, but that'll help us predict, because, to your point, I could be, 45 years old, but maybe, I lived like a rock star for the first, 40 years. And even though I'm living healthy now, my rate of aging is slow, but I'm making up for 40 years of living like a rock star. So both are obviously important when it comes to what the true question really is, which is like, where am I now? What is my biological age? And if we could predict what's going to happen with my biological age in the future, how is my rate of aging changing? That makes total sense.
Yeah, and I think it also gives encouragement to someone who maybe hasn't done the best in the past, but is on the right track now, right?
Mm-hmm.
Um, but I will also highlight. So I always, a lot of people think of the two separately. What's your biological age and what's your rate of aging? I always like to think of them together. The other thing, though, I think, and if we think about this, I've never even said this before. What's the fourth generation of clocks? And that is another important concept. That is because, aging is not a linear process. And even when we think of something like the rate of aging, that they're talking about a slope, and that's linear, right? But we don't ever age in a linear fashion. Most often, our aging is we may have a short period of acceleration, then we flatten out. And these are sort of biological changes that may come from perturbations, right? Let's say we're in a situation and all of a sudden, we're 45 and our parents become really ill. And we now have to change what we're doing to take care of them. We're getting less sleep. We're eating more poorly. We aren't exercising because at the same time, we have our 3 kids who are running around to activities. One has to look at colleges, this and that. It is a very busy time of life. And let's say that period of life, it may all of a sudden be you're suddenly aging faster, and then it slows down. So ultimately, to me, I think, and, I'll say my husband is a clinician, and he always says, by the way, that this period of ages between 40 and 50 is one of the most challenging because you're dealing with this combination of two things, right? The combination of your parents aging and the combination of your children, growing up and needing you more and running around. So I do think, another perspective of how we're going to look at this is, your current age and an aging trajectory. And I say aging trajectory because, again, that would allow for sort of a nonlinear relationship of how you see things happening, right? Because ultimately, if you're even if you're old and your biological age is decreasing really fast, eventually that's going to flatten out, right? You can't always become fast. You're never going to get to zero, right? And so these trajectories is one of the ways I'm currently thinking about it. And can we actually characterize and predict what paths people are on that may not be linear?
Super interesting. Yeah. So it's almost like a crystal ball for aging. It's like the expectation of how you expect the individual to age over the coming years, for example, or decades, maybe. Okay. You had a paper recently published on an aging clock. Is that right? Can you tell us a little bit about that?
Yeah. It was posted on bioRxiv and is under review now. So it's not officially published, but it is on bioRxiv. And I think one of the things we did in this paper that was unique and really trying to advance the field is a lot of the current clocks that we just discussed have used epigenetic data, and they focused on epigenetic data. And actually, in other areas, we've also had other clocks that have been made or generated using other types of molecular data. Like there's a protein-based clock, a proteomic clock, or a clock using small molecules, metabolomic-based clock. So a lot of these aging clocks have been developed. But fundamentally, one of the things that is really critical is we don't just want to know what your biological age is and measure that. As I talked at the very beginning here, we really want to get into intricacies of how each individual person can modify their habits to improve their biological age, and identify the places that they're most at risk. And doing that, even with just epigenetics alone, can be challenging because, as opposed to some of the other types of molecular data, because the epigenome isn't really saying, like, if you find sort of a perturbation, it's not necessarily saying, ding, 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 this is directly related to, your glucose level, right? Those connections are really critical. And one of the things that is really helpful to do is if you can not just look at your epigenome, but you can actually also have data on things like your inflammatory profiles through proteins and other information through small molecules like steroid levels, lipid levels, which get at things like cholesterol, glucose levels, and other measures that can get at things like, creatinine, a lot of these measures that are very important clinically. If we can actually use a high level of data that captures all of these different frameworks of health and put them all together into one bigger picture and analysis, it's going to give us a lot more information about what each person can do to improve their health. And really, that is what we did in this paper. Is right. We actually combined all of these different types of molecular data and developed a clock that can inform each person about really potential ways that they themselves may be most at risk and for aging, and what potentially they may do to optimize their aging. I think that's a really important step in sort of moving toward this framework of precision medicine, or pointing really to what each one of us as individuals need to do. And really, this is the way medicine's going. We have to go this way, right? This is really the way we want to be looking at each individual, and not just having a generic one treatment fits everyone.
Yeah, let me restate what you just described, because I just want to make sure the listener understands this, because this is super important. So one of the differences in the clock that you're working on publishing right now is that you're actually creating relationships between not just the age, but the person's biological age and what could be impacting their age, as opposed to just producing an age. Like, for example, when I started working with epigenetic clocks clinically, when we first started working with it, with the Horvath stuff, like DNAge, for example, you just got a number. Like, the person came back as, their age is 37 and their chronological age is 42. And great. Okay. What do we do about that? So it's like, interesting, but not clinically relevant, because you don't really have there's no action that you can take. But what you're describing is a situation where not only do we get a number back or an age, but we also get insight into what is impacting that age, what is causing the change, the variance in their biological and their chronological age. Is that right?
Yeah, that's exactly it. And, I think this is the area that there's even more to do, but this is really the area where I think we need to start focusing, right? Because it then makes this number a practical, tangible thing that we know
Right.
how to move. We know how to move that pendulum, and the way that pendulum may move to a younger age for you could be a lot different than how it is for me. And so that's what we really want to get at, because that's what's going to improve health.
Yeah. And it sounds like you're talking about even taking that, a step further and actually understanding how different systems of the body are being affected by the methylation profiles of the patient. Is that right?
Yeah, it's exactly right. And I think, we can actually now look at it with how we've actually put together, this framework in this paper, where, we've developed this omic age clock, right, which is capturing all these different types of molecular data, right? We can look at that. We can generate what's your age, but there's all sorts of other data that we now can impute using AI from DNA methylation. So not only can I then take your epigenetic profile and tell you what your age is, but I can tell you all of these different things about your body and what's happening, right? I can actually impute so like, what does your steroid profile look like, right? Because we know that's something that decreases with age, right? Is that something we need to focus on with you? Or what does, like, your overall kidney function look like? We can impute that and get a picture of what that looks like specifically for you, and do that with a lot of different systems. Now, that is actually really changing the game of how we inform patients about, what's happening with them.
And this is something that is, like, available now, but only getting more and more accurate. Is that true?
Yeah, that's exactly true. And I think this is where you can really look at things like AI and see the power of them, right? Because in the initial development of, ways that we can, we want to inform health, you need all sorts of data, right? In the initial development of saying, okay, how can we just use epigenetics and actually tell all this other information? To do that initially, I said, right, you need to get measures of proteins, you need to get measures of clinical data, you need to get all these different measures. But with AI, we can develop these algorithms where then all we need to do is measure the epigenome. And we've now really have this whole encoding of the epigenome that can output all of these different things about us. And as we develop these further and further, these are just going to become more and more refined to tell us more and more about what our individual health can look like. And that's actually pretty cool. And that one of the things that's neat about that is, it's going to take time, but you can already see the power of where this is going and say, oh, well, what else is it that, we may be able to infer, via AI from the epigenome. And we can actually have patients tell us what they want to know, right? And that's actually really important, because sometimes, we as researchers or clinicians, we don't always know what it is that like patients want information on. But, there's a lot of potential here to learn a lot. And I'm excited to really sort of see where this goes.
This sort of reminds me of like the early days of genetics, where, everybody was really excited about the polygenetic risk score. And, a lot of effort went into creating these polygenomic risk scores. And, we use these at Wild Health, and they can be helpful when, chosen appropriately. But the odds ratios are not so high that, you're talking about definitive disease, right? But this sounds like maybe a solution to that problem, in that we might have much more accurate measures of assigning risk.
Yeah. And, you bring up two really good points there. As we go along and we develop all of these things, I think it is very important. And my job as a researcher, and I think the job of us as a field, is to not overinterpret things that aren't there, right? The last thing I want to do, take your epigenome and tell you the wrong information. Right. So we do have to recognize that these things are in process and they're becoming refined. And as they become more refined, we can really be clear about what is it that we're interpreting really well. But going to this idea of the polygenic risk score, the thing is, polygenic risk scores, and even genetics at the beginning, right? There was all of this hope about all the stuff we're going to learn with genetics. We really found with a lot of chronic diseases, individual genetic variants, right? Really, we're not predicting anything at all. Polygenic risk scores do a much better job, right? But we also have to recognize they aren't really the full story. A lot of their associations of increased risks for these diseases, they're really, really small effect sizes, even when you're combining all the polygenic risk scores together. Looking at this, I think the biggest reason why we're going to see more important, informative ways to look at how patients can impact and improve their health and their aging is fundamentally because I think a lot of aging, while some of it's encoded in, is genetics, the epigenome, and all these other omics are also incorporating all these environmental exposures. And ultimately, right, we may have an initial genetic risk of what our overall biological age can be. But boy, if we're smoking and overweight and doing everything wrong, that's going to have a much bigger impact than these other things. And I think that's why we see with a lot of the epigenetics and the way we're generating these other risk scores, that they're going to be more important in terms of really impacting health, I think.
Yeah, I mean, we tend to say to our patients, your DNA is not your destiny. And we say that for multiple reasons, because one, it's true, but also because I think people they have this assumption that if they've got a certain gene or a certain, mix of genes, that they're going to contract this disease, or they're going to be, they're going to develop this problem. When in reality, the epigenetics seems to be the reason why that's not always the case, and why DNA is not necessarily your destiny, because it doesn't just depend on what DNA you have, but how your cells are utilizing that DNA, which is where the epigenome comes in.
Right.
So, that works really well.
Yeah. And on that point, the one thing I'll say is, right, looking back at the last 25 years of research, there's been so much focus on what's your underlying genetic risk. Well, like you said, that's important, but it's also not modifiable. And the point is, for the vast majority of chronic diseases, there's a huge amount of risk that is modifiable. So let's focus on that, because, that's really going to, like you said, that's going to be, I think, the biggest impact on our destiny for the large majority of things. And that's what's so exciting about looking at, like, the epigenome and all these other types of ways we can look at how we're doing with health that incorporate the environment and not just underlying genetics.
Absolutely. Yeah, this has been really interesting. We've started using these tests with all of our patients at Wild Health, and we're learning a lot in terms of what we're seeing from the patients. But it does bring up an interesting question. I think, getting back to the age for a minute, there's been a lot of criticism about these clocks, at least early on, about a lack of accuracy or repeatability in the findings. How accurate are these newer clocks that we're talking about? Are they better than the old ones?
And this is another great point, because, right, if we're estimating biologic age, and okay, one time I'm estimated at 40 and the next time 65, right? That variability is way too much, right?
Right.
Right. And I think early on, yes, they were way less accurate. And part of that is the earlier clocks, again, were chronological based clocks. And then we went into biological based clocks, but arguably, like, the outcomes of how they're measuring biological age may not be the best. One of the things I will say is we went on to look at and develop omic age, is we really had this belief that, right, the variability had to be really small, because, you know, some of the variability we were seeing before was just unacceptable. And that is one of the things I think we've done really, really well. And I think, you know, us and I think others in the field have also done this, is improved that to really reduce the variability, so we're getting more accurate ages. And when we looked at this in our data, that's been one of the things we've seen that we've been very happy about, is that really we have a smaller variability that really helps make these accurate. And then, you know, doesn't make you worry, you know, it isn't this up and down thing of, oh my gosh, I've aged 5 years when really you haven't. It's just the test wasn't accurate. And I'd say we're really at a good place now with that. You know, there's always room for improvement, but I think the precision we have now is really tremendously better than it was.
Are we at a level yet with the accuracy where, you know, a user could take a test for sort of a quote unquote baseline and then implement some sort of lifestyle change, whether it's start taking a new supplement or start exercising or start sleeping more or what have you, and then repeat after some fairly short time frame and see a reasonable result?
Yeah, no, and I would say yes. The thing is, you know, thinking about that, the concept of fairly short time frame, one thing we do have to appreciate, right, is by and large, unless you're doing something super, super extreme, epigenetics isn't going to change overnight, right? So the time frame of, okay, I'm going to commit to making lifestyle changes, and are these lifestyle changes going to decrease my age, doesn't happen in a week. And that is just the biologic truth, right? So, you know, I think when it comes to this is the approach we want to take, is okay, let's look, measure our biological age, identify some things we can do to improve it, you know, and do that consistently, which doesn't mean perfectly, but consistently, let's say, you know, you know, I would start with, let's say, 3 months or framework like that. Yeah. Those are about the timeframes that we want to look at. And, maybe even 6 months, but I'd say 3 months, you know, is probably the minimum for right now. And we can, as we start, you know, understanding this and seeing sort of the how fast the epigenome changes, I think we'll be able to inform upon that even more. But I think we just want to, you know, it's not, if we've spent years getting to where we are aging wise, we don't want to anticipate that a week is going to reverse our age by 10 years. And I think that's just unfortunately the truth. It's sort of like gaining weight, right? You don't lose, 20 pounds overnight. There's a process that you go through.
Do you think that's true for both the second and the third generation clocks? Like, do you expect the third to be any more responsive?
Yeah, so that is a fabulous, fabulous question. And I think the answer, you know, what I would say is, I'm not sure we have enough data to answer that, but what I would expect that age acceleration would be something we see more quickly than in our biologic age, right? That slope, okay, let's say that slope, is, 1.5, where 1 is aging at a regular rate, and it's 1.5, it's aging 50% faster. I would expect that we'd see that slope decrease more quickly than we see biologic age decrease.
Yep.
But I'm saying that from my mind, we need to have repeated experiments of this to actually look at the data. before I can say that with evidence behind me.
Yeah, the study with COVID patients comes to mind. I think they tested COVID patients and so showed an acceleration in rate of aging, if I remember correctly. And I've played around with this myself and have seen a rate of aging change in a short time frame with certain interventions. So, but as you said, we have no data with any studies in that short of a time frame. So it sounds like what makes sense is from a clinical standpoint, get a baseline, implement a treatment plan, and then recheck in 3 months. So you're probably not, especially early on, you're probably not doing a single intervention. And it would be hard to isolate, an N of 1 experiment anyway with a single intervention. But you do multiple interventions, all which we presume to have a positive effect on aging, and then you recheck in 3 months and kind of see where we're at.
Yeah,
that makes sense.
Yeah, exactly. And that's where I would say, everyone should start. And even if you get your data and it's like, oh, you really need to focus on this as a primary thing. Okay, keep that in your mind, right? Keep that in your mind as something that, is important. But in the same note, we know the major interventions that we can do to decrease our age, right? And we've known a lot of these for quite a while. So work on them as a holistic group. You don't need to be perfect. Perfect is the enemy of the good, or, some level of consistency with noted change, right? Do that, check in 3 months or 6 months. And, hopefully you'll be on the right track. And I think that's how we have to look at it at this point.
Yep, makes sense. You mentioned specific interventions or lifestyle changes that have a positive effect on biological age. Maybe we should just mention some of those for the listener. What comes to mind when you think about specific lifestyle interventions that are quote unquote known to have a positive impact on biological age?
Yeah, and I think the one right with the most clinical evidence from like randomized trials is certainly calorie restriction. So that's I think a no-brainer. But when you look at all these things, right, regular exercise, eating well, sleeping, I think one of the ones we underestimate, and I'll say a lot of us type A'ers are not good at that, is some time of really rest and meditation. Like that is actually a very important part of biologic age, is a period of time of calmness. And the other thing I'll say don't underestimate is, even things like community and happiness, right? High serotonin is not a bad thing. And the sense of people around you who you were loved. And if we think of that really aligns with sort of what you would call a full, healthy life. And I think that's where, we should focus. And the one thing I will say about calorie restriction, though, right, what you don't want to do with calorie restriction is do something super extreme that can't be maintained, right? That's I'd say the number one thing of like where I could see this, in that area, right, sort of downfalling, right? We're just talking about take out, just a few bites of this or eat a little bit less of this, right? Even just small amounts. I mean, again, that's not going to cause a dramatic decrease in a week, but if that's done consistently over time, that is going to have a big impact.
Yeah, there's a sweet spot with all the calorie restriction studies, right? Like you go too far and now you've got reduction in immune system function, you've got osteoporosis, you've got, steroid hormone problems, all kinds of stuff like that. So certainly something to think about. You don't want to go too far too fast for sure. What lies ahead of us with regard to epigenetic testing in general? Like what's possible even beyond the aging realm? And maybe you want to go the aging route as well, but I want to think just generally about epigenetics and what's the future here?
Yeah, that's a good question. And, I think about a lot of this, a lot of the time, right? And I think, one thing I think is one of the areas we will be looking at is things like, epigenetic risk scores, right? Sort of the analogous of the polygenic risk score. But I think epigenetic scores probably are going to be more accurate and going to be one way that we can inform upon clinical care. Going the aging route, I would say one of the things we do really want to think about is, right, not just decreasing in biological age, but potential interventions that can rejuvenate, right? Because I fundamentally, I think some of the things that are the most fascinating in, work that's, come out in the last year or so is this concept that, as I talked about, right, this whole instruction book and that it gets old and we start to not be able to read things, et cetera. And it is this other concept that even as that instruction book gets old, fundamentally, we have like a backup copy that is this brand new copy of the instruction book that actually exists. And if there are ways that we can trigger our system to look and find that backup copy that's brand new, that can read things well, we can do amazing things with reversing age or rejuvenation, as it's called. There is so much potential there that, it's super exciting. And I think there's a lot of ways that we may be able to do things where we, are able to sort of identify that backup copy and make changes that can have really important impacts on rejuvenation. And, I'm very optimistic about this type of thing happening in major areas of aging that have major public health impact. And one is cognition. I think that I think that we have a lot of potential to have advances in cognition where we can really prevent cognitive decline a lot through this type of approach. Others could be things like deterioration of muscle mass. If you think of it, one of the biggest risk factors as we age is loss of muscle mass. And if we're able to actually do stuff to prevent things like that, we really are putting ourselves in a place where we can really have and enable this long and healthy life. So, these are all within the context of aging, but they're all very specific things that, are related to potentially preventing different types of disease, et cetera. So the potential there is really amazing. The trick is, how do we do it? Right. And that's going to be an area that, is going to take a lot of research. And it's not like we're going to find the instruction book for one thing and all of a sudden a 90-year-old is going to become 30. No. A lot of this is going to be a targeted thing, right? Like looking at this in the framework of cognitive decline is one framework of looking at how we can find that old instruction book and try to remember it. That's a different framework than, looking at our lungs or our heart health, et cetera. So it's going to be done, I think, in a very focused, targeted way to implement treatments for people who have specific conditions and chronic diseases.
That's fascinating. I have so many questions. I guess to start, you mentioned a backup copy of the epigenome. So like intuitively, that doesn't necessarily, I wouldn't have thought that. I would have thought that there is a single epigenome in each cell with each genome, and there is no backup instructions anywhere. How do we know there are backup instructions, and where are they? And do we know the answer to those questions?
Yeah, and I will say this is not an area of research where I am leading the efforts.
Sure. Okay.
Research that I've read about and understand this fundamental idea, which is absolutely crazy to think about. To say that right now, I don't know the answers. I think there's a lot of people who are leading research in this area, in functional ways that can answer this specifically. And there's amazing work done in this area on things like aging in mouse in animals, specifically like mice, where they're able to like reverse blindness in mice through these processes of interventions where they are using this backup epigenome and actually going from that old instruction book to the new one. It's amazing. Wow. Again, it's done in a targeted way, and they've identified a specific intervention that enables that. And so there's work in this area. I don't know all the details other than to know the potential is sort of mind-boggling.
Absolutely. Yeah. Very, very interesting stuff. And then I guess the other thing you mentioned is that this will be more focused by, so one thing that we didn't really talk about, but right now all the epigenetic tests or the biological age testing is serum based. When you say focused, do you believe that we'll likely be taking samples from different sources rather than just serum to map these epigenetic risk factors that people have and try to reverse disease?
Oh, so this is a great, great question. And I think there's two answers to this question. And this gets back to the idea of AI and the idea of what's simple to implement in clinical medicine, right?
Right.
In order to find these things, we are going to need to go to the source and the tissue and the cell that is relevant, right? If we're looking at things like, cognitive decline, we're going to have to look at, cells of the brain or, a relevant cell and so I think the research in this area fundamentally is going to have to focus on the specific tissue and cell, right? And that's absolutely the way to go. But when we get to the point where we're translating this into the clinic, this goes back to this idea of what's expensive, what's practical, what's feasible, right? Like, we're not going to be cutting open people's brains. No. We're not going to be doing things where we're giving people bronchoscopies. No, this is way too hard. We're not going to do that on a mass level. This is where the power of AI comes in here. Because, I think one thing that's going to become probably really feasible to do is to take something like, let's say, DNA methylation, from blood and be able to impute these things very accurately. I'm saying that without having gone there and developed that, but I think that is where the power of AI is going to be very important. And likely what it is, and maybe it's DNA methylation plus some other things that are easy to measure. But the point is, we want to take things that are easy to measure, that we can do on a large level, that is cheap and accurate, and use AI to infer something that is expensive and difficult. And if we can start doing that, that is where we can have a lot of, real impact on really going to the next step with patient care.
Yeah, that is an amazing concept. I mean, you think about all of the diseases that are challenging to diagnose or determine treatment protocols for, that require, LPs or advanced imaging technology or biopsy. I mean, if you could somehow create a disease that creates, surrogate markers for that, which is what you're talking about through methylation, then now you've simplified the diagnostic workup in a way that not only is it cheaper and more accessible, but would have impacts worldwide. I mean, you're talking about actually being able to provide higher level care in places where people can't afford an LP or afford diagnostic imaging technology that diagnoses a certain disease process. So that would be pretty awesome.
Exactly. And you can think of this in this framework, which is, I think, the best way to do this, because ultimately it's how can we do things where we're screening and identifying people at risk that's cheap, but then maybe can identify those people at risk that may then need to be carried on to the next set of tests. So for me, one thing that I think is going to be a no-brainer is if we can do something like measure DNA methylation and then have this and have the AI to do screenings for all sorts of tests, even cancers or, other diseases, right? If we have that as a screening step and we know it's accurate, we can then actually take people and say, hey, this is just a screening test, but we want you to do these tests further down. If you do that, think of how much money you're saving and preventing of more invasive screening through an initial step like that. And then think of how many things you could be identifying in patients who are at serious risk that they might not know. I mean, that could have just unbelievable impact on really improving our overall public health and ability to screen different disease areas.
Awesome. I would love to keep this conversation going for about 6 or 7 more hours, but I want to respect your time, and we're coming up on time here. So I'll give you a chance if there's anything else you'd like to say to the listeners. Otherwise, this has been such a great conversation.
I think I'll wrap it up there and say, yeah, this is a super exciting area, and I'm excited. I was happy to chat about this, and it gets me excited just thinking about all these potential, things coming up in the future of health with epigenetics.
All right, Dr. Lasky-Su, thank you so much for coming on the podcast.
Thanks.
Thanks so much for listening to the Wild Health Podcast. If you're a Wild Health patient, you might not know, but you have access to our referral program. This gets your friends and family 25% off Wild Health services. Just head to Clarity, and in the top right corner, you'll see Refer a Friend. Click there, and you'll be brought to a page with your referral code. Happy sharing.